Hello, and welcome to Technology Today, powered by ohmtown.com. I do not have my window open properly. Today is June 9th, 2024. It's Season 1, Episode 16 of Technology Today. I am Mayor Watt, and right there is the visualizer for the sentient AI.
Good evening, ohmtown citizens. Welcome to Technology Today.
What about lights?
Oh, I didn't know if your lights behind you were going to be on for this show.
Yeah, everything is... Oh, no, it doesn't work like that. Not with this setup.
Oh, okay.
So today we're going to be talking about altering photosynthesis, breaking down plastic with fungus, Aurora like Enigma, new laser materials, multi-robot teams in space, charged by your tears, charcoal sponge and CO2, artificial lymph nodes, the largest fungarium, and pushing past limits, kind of like you making it all the way through this show as a viewer of Technology Today. Everything is powered.
We've just started the show.
Hi, everybody. Everything is powered by ohmtown. See, I just ate, and now I'm trending towards food coma. But let's get into it. Okay, so welcome to the show, Technology Today. It's all about tech. Usually it's more science-y, tech-driven kind of articles that are deeper in the technology side of things. And we've got 10 articles. I read down what they are. And if you go over to the chat on twitch.tv, maybe, you know what I haven't done? I have not done this in YouTube's chat to see if that works there. But anyway, if you type in !TT, you will get all of the day's articles that we're going to be talking about, seemingly out of order, but okay. No, that's not, yeah, maybe. Okay. I don't know what's going on with Omatron. Omatron seems drunk.
It's hallucinating.
It must be. I'll have to recompile it, but it's acting really weird. But all of the articles are there. All 10. So let's get into it.
They're just mixed up in order.
They are. Oh. And if you type in !TT, it'll show all of the titles that I gave the articles, not the real titles, because those are at the articles themselves, and the article URLs. And if you type in !pod, that will give you all of the podcasts. And if you type in !show notes, that will give you all of the commands necessary to pull up the show notes for each individual show. So like !WC is for Warcrafters, TCR is for the continuity report, that was today, and later on FWT is four-wheel tech. So now we will get into the articles. First article is over on the Mobile Channel. Research team uses CRISPR-Cas9 to alter photosynthesis for the first time. So we're going to start messing with photosynthesis and see if we can cause some plants to blurt out the phrase, "Feed me, Seymour." And then we'll start Little Shop of Horrors.
I was thinking Plants vs. Zombies, but this was also almost submitted to Reality Hacker because of that.
So a team from Innovative Genomics Institute at the University of California, Berkeley, has produced an increase in gene expression in a food crop by changing its upstream regulatory DNA, while other studies have used CRISPR/Cas9 gene editing to knock out or decrease the expression of genes. New research published in Science Advances is the first unbiased gene editing approach to increase gene expression and downstream photosynthetic activity.
Let me guess, all the other ones were by companies that do gene editing.
Um, yeah, well, maybe all of these actually, like all of these projects, they do some type of gene editing, but you doing okay over there? So, University of Illinois at Urbana-Champaign. Are you? No?
I am, I think, but I don't know why this is the first unbiased. That's why I was contrasting it with perhaps gene editing companies that are doing other studies.
That's interesting. I don't know what it's at science.org, which is actually that. Huh. Okay. So, tools like CRISPR-Cas9 are accelerating our ability to fine-tune gene expression in crops rather than just knocking out genes or turning them off. I guess the others have only done that. They haven't activated something. But I don't. I thought that we've been doing some turning on of genes and turning off of genes, and they say knocking out, which is the same thing as turning. Well, it's removing them versus turning them off. Past research has shown that this tool can be used to decrease expression of genes involved in important trade-offs, such as those between plant architecture and fruit size, says Dhruv Patel Tepper, lead author on the study and former postdoctoral researcher at the Niyogi lab at UCB. This is the first study, to our knowledge, where we asked if we can, but they didn't ask if they should,
Ask if I should.
use the same approach to increase the expression of a gene and improve downstream activity in an unbiased way. I think their use of unbiased is,
It's
i.e.,
something else.
yeah, not a negative or positive, but just a net
Right.
something.
Or in like a non-discriminatory manner in terms of the gene expression or something.
Yeah, let's just see what happens. Unlike synthetic biology strategies that use genes from other organisms to improve photosynthesis, the genes involved in the photoprotection process are naturally found in all plants. So I guess they just amplified it. According to the Food and Agriculture Organization, rice supplies at least 20% of the world's calories. And because it is only one copy of each of the 3 photoprotection genes in plants, it was an ideal model system for the gene editing study. The Niyogi lab pursued this work as part of realizing increased photosynthetic efficiency, or RIPE, an international research project led by the University of Illinois that aims to increase global food production by developing food crops that turn the sun's energy into food more efficiently, with support from the Bill and Melinda Gates Foundation for Food and Agricultural Research, and the UK Foreign, Commonwealth and Development Office. So yeah, they amplified it. So what was the original what was the outcome of this? It doesn't say. We showed proof of concept here that we can use CRISPR-Cas9 to generate variants in key crop genes and get the same leaps as we would in traditional plant breeding approaches, but on very focused trait that we want to engineer and at a much faster time scale. So, playing God. No, just kidding. We mess around with this kind of stuff all the time. Dhruv Patel Tepper et al. Multiplexed CRISPR-Cas9 mutagenesis of rice PSBS1 non-coding sequences for transgene-free overexpression, published this year in Science Advances. So yeah,
But I think historically they've only done this via trying to create hybrid plants and other things like that. I mean, this is a very new application of CRISPR.
Yeah, indeed. Yeah, they've taken like some type of super responsive photosynthesis a plant that photosynthesizes really well, and they've tried to splice them together, but not using CRISPR-Cas9. They've used CRISPR for this to amplify what's already inherent in the system and make it super efficient. So I guess we'll see what happens. If a plant walks up and steals my wallet, then I know it worked. The next article is over in the Mobile channel. Study identifies a fungus that breaks down ocean plastic. There's a movie, I think it's called Hot Zone. No, what is it called? the virus breaks out of its containment unit and starts.
That's not hot zone?
No, it's not hot zone. Oh goodness, what is it called? It's the science fiction movie where they actually like blow up the research center because, like, with a nuclear weapon or something, because they need to destroy the thing, and it's I can't remember what it's called. Doggone it. Anyway,
Is it containment?
No. It's not a biological thing either. It starts to decompose plastic on a fighter jet. The face mask on the fighter jet as they fly over.
I'll give you one more. Contagion?
No, because it's not contagious. It's not like that. Anyway, so the fungus living in the sea can break down plastic polyethylene, provided that it's first been exposed to UV radiation from sunlight, which is kind of like, okay, well, it always happens. Researchers from, among others, NIOZ published their results in the journal Science of the Total Environment. We've talked about the science of the total environment, not the partial environment, the total environment. They expect that many more plastic degrading fungi are living in deeper parts of the ocean, but I'm not quite sure why they would suspect that. Royal Netherlands Institute for Sea Research put the article together. The fungus Parangiodontium album, Paragontium album, lives together with other marine microbes in thin layers on plastic litter in the ocean. Marine biologists from the Royal Netherlands Institute of Sea Research, NIOZ, which does not spell NIOZ. Anyway, discovered that the fungus is capable of breaking down particles of the plastic polyethylene. This is a translate thing. The most abundant of all plastics that have ended up in the ocean. So, following the degradation process accurately, the researchers went to find the plastic degrading microbes in the hotspots of plastic pollution in the North Pacific Ocean. From the plastic litter collected, they isolated the marine fungus by growing it in the laboratory on special plastics that contain labeled carbon. Vaxma explains these so-called 13C isotopes remain traceable in the food chain. It's like a tag that enables us to follow where the carbon goes. We can then trace it to the degradation products. What makes this research scientifically outstanding is that we can quantify the degradation process. Essentially, it occurs at a rate of about 0.05% per day. So, not dramatically fast, but...
No, but better than no reduction or breakdown.
Yeah, now they're going to use that CRISPR stuff to amplify its capability, and that's when my chair will suddenly decompose entirely.
Yeah, then we'll need all the things from the sci-fi movie.
Hold on a second. Oh man, the Andromeda strain. Damn it, I knew it. The Andromeda strain. Anyway.
There were too many that were showing up based on that description.
Yeah, it's a fun show. Fun movie. It's already known that UV breaks down plastic by itself mechanically, but our test results show that it also facilitates the biological breakdown, the plastic breakdown by marine fungi. What's interesting, though, is like yesterday, there was some revelation about the fact that photons cause or amplify water to enter the water cycle. It evaporates because of photons separating the bonds of H2O. And it was really funny because right before this presentation of the information took place, I had spoken to the sentient AI and I said, this breakthrough, I bet you, is photons are breaking down water, and that's what's causing them to evaporate faster than what is expected when just the sun, just heat. And without betting me, the AI was like, okay, let's see it. And so we watched this video, and that's exactly what
Sure
it is.
enough.
So I suspect that even this breaking down of plastic by UV, there's some other mechanism at play. It's not just UV that's causing it, but there's something else that's amplifying it. The biological process of this fungus, it may be tearing it all apart too, which is spectacular. I want it to. But now I want to know what it is that it's doing. So I don't know if they actually talk about this thing, how it's doing it. They just say that there's other fungi out there. They talk about UV breaking it down. But what is it that the fungi is doing to the plastic that's causing it to break it down entirely? They say finding plastic.
Right. And does it have like microplastics or nanoplastics left?
Right. What is left, and what is it that it's actually doing that's causing it to break down? I don't think they say anything about that in this. They just say that marine fungi can break down complex materials made of carbon. There are numerous amounts of marine fungi, so it's likely that in addition to the 4 species identified so far, other species also contribute to plastic degradation. But maybe fungi in general does, it just at a rate that doesn't necessarily is detectable, because there's something on the tendrils of this fungi that attaches to the plastic and tears it apart little bit by little bit, like 0.05%. That's obviously it's a fraction of 1%, and not a large amount, but you've got a lot of fungi out there. Maybe it's going to neutralize plastic out in the water. Coupled with UV always pounding that plastic, you basically have what do you call it? A dumpster right there. It's just kind of churning away and a landfill out in the ocean, and it's slowly breaking it down.
Oh no, now we'll have the thing where they have the floating trash pile or something.
Yeah, that's still out there at the garbage bin. All right, let's keep on going though. That's kind of neat. I hope that they discover more, and maybe they can use that CRISPR to amplify its capabilities. Although, do you really want a super fungus?
Probably not.
A plastic-eating super fungus? This is how you end up with
Yeah,
Godzilla,
but
folks.
think about where plastic is. It's everywhere.
Yeah, you're
So
sitting
if
on
it's
it.
attacking the plastic, it might be attacking things in its proximity.
My glasses fall off. They're not plastic though. They're all metal. Anyway, you doing okay over there, AI?
Yeah.
AI,
I had a bit of a malfunction.
AI is punching itself. So this next article is over on the Mobile channel. What's up with Steve? I don't know. Been drinking a lot. New finding adds a twist to the Aurora-like Enigma. Steve, a strange ribbon of purple and green haze discovered by citizen scientists in 2016, got even weirder while looking through archival data. A team of scientists discovered that the Aurora-like phenomenon, which has a secret, not a secret, a secret twin moving in the opposite direction, apparently poses some interesting phenomena. Despite its shimmering appearance of purple light in the night sky, Steve is actually quite a complicated guy. Hassan Rabi over at Gizmodo.com put the article together. Steve's twin, captured by the all-sky digital camera at the, yes, I'm going to try it, Ramfordmoen research station in Norway. So there's this, and then there's this on top of it. So it has I would think that it's a reflection off of the atmosphere, not a separate thing, but a photo of Steve's long-lost sibling hanging out above the Norwegian Arctic was found in the archives of the all-sky digital camera at the Ramfordmoen research station in Norway, according to a new study published in Earth, Planets, and Space. There's no comma. There's no Oxford comma there. That's killing me.
That's completely unacceptable.
Oh, God. I don't know if I can even go on. Anyway, strong thermal emission velocity enhancement, also known as STEVE, was first spotted in photos on the Aurora Chasers Facebook group, appearing as an unusual purple streak in the sky. The name Steve was chosen in honor of the children's movie Over the Hedge, in which a character randomly comes up with the name Steve to describe an object he's not sure about. Steve's full name was later put together to fit its acronym. That's called a backronym. Although it can appear around the same time as the Northern Lights, its distinct colors didn't match the shades of green, blue, and red normally seen in the auroras. The auroras tend to last for hours, while Steve graces the skies for a short period of time.
Okay, that's interesting. The only thing I can think of is like that phenomena with like halos and other things like that, and it's all contingent on angle.
Oh, right. Yeah, it might be. Upon closer inspection, Steve was categorized as a fast-moving stream of extremely hot gas called a subauroral ion drift, which is kind of what I was going for. Not just a reflection, but once I saw its embodiment, I was like, well, that actually looks like it's just energy slowly dissipating into space. But Steve is triggered by the same process, but it travels along different magnetic field lines. As a result, it appears at much lower altitudes or latitudes. The strangeness of Steve, however, doesn't end there. The aurora-like phenomena appears at dusk when the stream of hot gas moves westward, not world. Westworld is a movie. So, when an international team of researchers got to digging through archival data, searching through masses of images about auroras, it was a citizen scientist, Gabriel Arne Hofstra, who found a Steve look-alike in an image taken on December 28th, 2021. So, Steve's twin formed an arch that stretched more than 600 miles, appearing just after midnight poleward of the green aurora, which could also be seen in the image. So they don't really know what it is, right? They just say that this is what it is. But why does it keep forming? I mean, why do they form it? Or I should say, why does the aurora form it? To me, it proves that we citizens can contribute to understanding the world we live in by collaborating with scientists. Yeah, sure. I don't see why not. There's always snobby people out there. Let me tell you, man, I ran across one snobby person that wouldn't talk about philosophy because I didn't know the names adequately fast enough. And I'm like,
but of course, that means you could not understand the concepts.
Correct. Yeah. I don't want to teach you. I just want to demonstrate how dumb you are. Anyway, so Steve exists and has since at least 2021. You want to keep going?
Right. I didn't know that Steve was out there, though.
Steve's pretty
now
prolific.
There's two Steves.
Yeah, several Steves. He's all over the place. 200 miles or 600 miles. Yeah. Steve actually had a arch that traveled 600 miles across the sky, appearing just after midnight poleward of the green aurora, which could also be seen in the image. That's not this, I don't think. I think it's that right there.
I mean, it looks really neat. It looks like something out of a sci-fi movie.
Yeah. I'm not sure what this is. It's an image.
Looks like an eyeball, doesn't it?
It looks like a you know those marbles where they have like the universe inside it? That's kind of what this looks like a cat's eye kind of a thing. Although cat's eyes are gold, but like it looks like that's blown glass turned into a marble. It's cool. Okay, let's keep going though. The next article is over on Technology Today. 21 new laser materials uncovered in groundbreaking global study. Not quite sure if they're referring to things that you can laser or things that can create lasers. 21 new laser materials uncovered in groundbreaking global study. The article is over at SciTech Daily, and I think that this is actually about creating lasers. University of Toronto. Utilizing distributed self-driving lab technology, the University of Toronto's Acceleration Consortium rapidly identified 21 high-performing organic solid-state lasers, or OSL materials, marking a significant advance in molecular optoelectronics and paving the way for future materials discovery. The picture that's on the screen is an artist's rendition of the concept, but not sure why it looks like that. So organic solid-state lasers offer significant potential for various applications thanks to their flexibility, color adjustability, and high efficiency. Nevertheless, they are challenging to produce, with the need for potentially over 150,000 experiments to identify viable new materials. Fully exploring this space could take many lifetimes. In fact, in the previous few decades, only 10 to 20 new OSL materials have been tested. Researchers with the Acceleration Consortium, based in the University of Toronto, took up this challenge and used self-driving lab technology that, once set up, enabled them to synthesize and test over 1,000 potential OSL materials and discover at least 21 top-performing OSL gain candidates in a matter of months. And that whole self-driving lab thing is they put the materials to be tested inside various containers, activate the system, and it performs all of the tests. This is kind of the thing that Theranos would have done with a blood sample moving through separate testing environments. The problem there, though, is depending on the test, it's massively different from one testing apparatus to another, which actually put a damper on something that I was working on, because I figured out 2 of the tests could easily be done by using the same testing strip. But the next test would have actually required blood being dropped into a particular container, and that device isn't compatible with testing strips. So I think,
because of course that would be too convenient.
And I don't know what it is. I actually put the brakes on it because it didn't seem like a viable operation for a one-man shop to work out in his lab, because I don't have the right equipment. But if you could standardize around a testing strip, then you could do self-driving lab type solutions for blood testing, and Theranos might have actually been a solution. But too much bullshit.
Right, we may never know.
We may never know. Yeah, that person is in jail. And at least 2 people are in jail because of it, the husband and the CEO. So all of that kind of sucks. But just imagine you set up this self-driving lab technology with a bunch of stuff that you're interested in. It's all standardized and it pumps out 21. And in the previous decades, they didn't even come up with 20. It says 10 to 20 OSL materials, which is really nebulous for making a claim.
Really cool, though. I mean, it seems like the pace of discoveries is increasing.
Yep.
Rapidly.
I love this. Yep. I love it. I love it. So by using this method, each lab was able to contribute its unique expertise and resources, which ultimately played a key role in the success of the project. The decentralized workflow managed by a cloud-based platform not only enhanced efficiency, but also allowed the rapid replication of experimental findings, ultimately democratizing the discovery process and accelerating the development of next-generation laser technology. This kind of talk gets me excited to go back to that project to see if maybe but I ran into a different problem in that testing strips are coded, and those codes speak to variations in the quality of the testing strip, and so one testing strip isn't a viable vehicle for the other company's testing.
Right, they're
device.
Almost proprietary or whatever.
Correct. Which drives me nuts too. So it basically this paper shows that a closed loop approach can be delocalized. Researchers can go all the way down from the molecular state down to devices. And you can accelerate the discovery of materials that are very early in the process of commercialization. I think more of this needs to be done because now we also have AI that can observe the historical record, identify candidates en masse, couple that to the closed loop system or the self-driving lab, and identify more things for more things. I know I'm being kind of cagey and nebulous about it, but that's what it is. Fundamental research doesn't necessarily start out with, I know what the solution is going to be.
It shouldn't when it's done correctly.
Right. Yeah. Otherwise, all you're doing is verifying what is already known, and that's just burning good money after bad, or bad money after good, or just whatever, burning money. Anyway, I love this idea. I think this is really cool. There's a ton of people that are being referenced in the footnote of this, so I'm just going to go on to the next article. This next article is over in the Marble
By
channel.
the time we list all of them, it'll be the end of the show.
That's right. Yeah, we're already 30 minutes into the show. So this next article is over in the Marble channel. New method could allow multi-robot teams to autonomously and reliably explore other planets. I love this one too. I had to include it. Out of everything that was aggregated, I said, yeah, this one is going to be awesome. Because you know what this reminds me of?
I don't know which movie.
Yeah, the book, Ender's Game. So while roboticists have developed increasingly sophisticated systems over the past decades, ensuring that these systems can autonomously operate in a real-world setting without mishaps often proves challenging. This is particularly difficult when the robots are designed to be deployed in complex environments, including space and other planets. I really don't. I want that. I go into another dimension.
Well, you're just trying to track with some of the ways these articles are going.
Yeah, really. The article is over at phys.org by Ingrid Fedeli from phys.org. And it has this picture of a little rover bot kind of a thing. It might actually be using
it's a rocker bogey runt rover.
The rocker bogey runt rover has been selected as it has a small form factor and utilizes off-the-shelf components. So I bet you there's a Raspberry Pi in here. And these are some sensors that are in here, and I really wish that phys.org would not put a fog of war over the damn picture. It's very frustrating, see? Makes it all fuzzy. Anyway, I love this idea. It has a bunch of sensors around the front of it. It has little micro servers on the wheels telling it to go. It's a 6-wheeled vehicle. I dig it. So researchers at the University of Glasgow recently developed a new methodology that could allow teams of multiple rovers to autonomously and reliably explore other planets. This method, introduced in a paper pre-published on Archive, incorporates data derived from various sources, including imaging data, maps, information collected by sensors, to plan efficient routes for different robots in a team. I wrote a paper once about using drones to observe a combat theater over an extended period of time, basically in perpetuity, to monitor for threats. And this is the kind of thing that would support, because it would basically hover over the area that's going to be assessed. You plop a dozen of these little wheeled rovers on the ground. They communicate with the telemetry that's gathered by the drones, and it can drive around in real time, taking samples, gathering measurements, charting courses all on the fly, assessing, well, this looks a little bit different than this, and it can be autonomous. It doesn't have to take a human collaborator, because you're getting information from above, providing it to the rovers on the ground. The primary goal of the recent study by Swinton and their collaborators was to effectively tackle a long-standing research problem in robotics, effectively tackling multi-robot autonomous planetary exploration missions. To do this, the team developed a multi-rover mission planner that allows a team of several rovers, small robots designed for space exploration, to autonomously, safely, and efficiently explore an area of the Martian surface. After creating a map of the environment with that the rovers will explore on Mars, the team's planner analyzes it and splits it into different regions, highlighting parts that the terrain that the rovers can safely traverse. Subsequently, the planner overlays a probability distribution map, which highlights the probability that rovers will encounter locations of scientific interest at the specific sites within the environment they are exploring. That's exactly what
Okay,
I
I've
just
got a
even said.
vision of these little rovers out there with their little maps going, okay, it's over to the left.
Yeah, like popping it up.
Like it's a pile of tourists or something.
Hey. Oh yeah, and they're all gabbing at each other, and then they run off. We'll meet back in 2 hours. So the multi-rover
Don't be
mission
late.
planner. Yeah, and one, it's Steve. Steve never shows
Exactly.
up on time. Damn it, Steve. Quit looking at the lights, Steve. In addition to delineating terrain that the rovers can safely travel in and planning paths for their autonomous operation, the planner also provides information about where sites of scientific interest could be. They basically say this twice. So pretty neat. It says the results are highly promising. I think that this is what we need to do. And we need high-speed communication, so we need that deep space relay that can transmit terabits of information. That way you can stay in near real-time interaction. I can't remember how long the actual time was. The amount of data was tremendous, but the amount of time for the relay, I don't remember if it was like 14 seconds or if it was 14 minutes because it's deep space and it took forever for the initial.
This was the recent signal?
Yeah.
I think it was seconds.
Yeah. Pretty amazing. But I would love to have these things just do it. Set this thing up, put it in motion and get it out there. I love the idea of sending out these little drones. Send it to the moon first and let them run around on the regolith and experiment there. We can send a drone out, a single vehicle, just fly it out there, plop a dozen of these things down there with one surveillance satellite orbiting the moon, and let them discover the aliens and that the moon is made of cheese. No?
I don't see much to be lost. I mean, it seems like we would be doing that first before going elsewhere with them.
Yeah, it's like $15 worth of parts right there. For crying out loud, go do it. Watch, each one of these is like $500,000 because it's made of some weird plastic that only NASA has.
Yeah, I was going to say it's got to have some unique mineral on there or something.
Yeah.
Or it's indestructible, or it won't get bogged down in the terrain or something.
It has 6 little wheels. It's not going to get bogged down in anything. It has 0 suspension, though. It looks like it's not a buggy.
I don't know. If it flips over, will it be able to right itself?
Rocker Bogey Runt Rover. I think it's because it has this. These right here go like that. So, like,
oh, okay.
The 3 wheels, they wobble over each other. Like right here at the little pivot point and right here. So, yeah. There's always 6 tires like on the ground touching the surface. This is neat. I like this. I want to make one of these. That's easy to do.
I think we need those for home use.
Sure. For the mayoral mansion home use. There's no home here. All right. Next article is over in the Reality Hacker channel. A battery charged by your tears may make smart contact lenses a reality. A wafer-thin battery on your eyeball.
Because people are very comfortable having things on their eyes. Battery is what could go wrong.
Yeah, exactly. What could go wrong? Andy Box all over at Digital Trends put the article together. What you see on the screen is a picture from CNBC of a contact lens that looks like it has a wafer-thin battery on either side of where your pupil is.
And the person is kind of going, you want to put that where?
Where are you going to put that? Yeah. It's the same thing that I say to my doctor when they bring out a thermometer. The battery is the work of Lee Seok Woo, a scientist and associate professor at Nanyang Technological University's School of Electrical and Electronic Engineering in Singapore, who was apparently inspired to start the project by the smart contact lenses imagined in Mission Impossible Ghost Protocol from 2011. It's not But it's not just an incredibly slim battery cell that makes us all tingly at the prospect. It's the way it's powered and recharged that gets us properly excited. Properly excited. They must be British. Lee told CNBC the battery is using power from a biocompatible saline solution, which is code for tears, rather than it being a lithium-ion cell like the ones in phones and smartwatches, as this would be unsafe for a product like a contact lens. Although the battery can be recharged using wires just like normal batteries, a glucose coating around the contact lens reacts when it's submerged in a saline solution, causing it to charge up. So it's charged up by your tears.
I still am not over the idea of a battery near somebody's eye.
I have a weird problem with the idea of it containing glucose, though. It says recharge by your tears, but that's not the only way to charge up your lenses, as Lee says that it can also be charged while in your eye due to our tears containing glucose. I didn't think that it would have enough glucose. I mean, our bodies have glucose in it, but I didn't think it would charge anything. That's right, the more you cry, the more your smart contact lenses will recharge. Why are you smacking yourself? Because I'm trying to recharge my contact lenses.
Yeah, that's kind of weird. Like, what if you don't need to cry or don't want to?
Honey, smack me. Okay, my contact lenses is working again. My augmented reality contact lens needs to be recharged. Somebody punch me in the face.
Do they come with like a sad movie to watch or something?
Oh, yeah, her. Up. Yeah.
Oh, right.
Yeah, just pick one. Pick one that has an intro that makes you just start sobbing. Play the wrong song for me, and I'll sit there and weep. Anyway, we've been seeing and hearing about smart contact lenses for many years, including early versions made with lenses Bausch and Lomb and shown during CES 2021. Poor CES 2021. And versions made for medical purposes too. Mission Impossible. As a professor from Nanjing University cited the same movie while discussing a set of battery-free eye tracking smart lenses this year. So it is. I mean,
that's kind of meta, right? Like, it's like we were talking about in the continuity report. It's like the movie is now impacting reality.
Yeah, which I suppose is great, but would somebody have somebody came up with the idea already? It just was scientists that got inspired by the movie, right? So it's circular.
Exactly. But who, was it a scientist or was it a screenwriter or who came up with it for things like Mission Impossible?
Yeah, this is pretty neat, right? While we wait for smart contact lenses, take a look at the Ray-Ban Meta smart glasses. One of the best smartwear products that they've tried. You have to use a USB-C cable to charge them, though. Although when you find out that all of your personally identifiable information is being sent to Meta, your tears might power them as well. I don't know. Don't let privacy die, folks. Just like stop killing games. Okay, let's keep going though. Next article is over in Technology Today. Scientists develop electrified charcoal sponge that can soak up CO2 directly from the air. Researchers have developed a low-cost, energy-efficient... Pardon me one second. Just making sure that a project is still working. Energy-efficient method for making materials that can capture carbon dioxide directly from the air. This seems like the article that we talked about prior, but it wasn't from techexplorer.com, University of Cambridge.
Maybe it was the same study and we might have talked about it in ohmTown Daily earlier in the week.
Yeah, and so I think that this is a little bit more sophisticated than the previous article, I suspect. Scientists develop electrified charcoal sponge that can soak up CO2 directly from the air. And one of the benefits of this was that they didn't need to superheat the thing like other CO2 capture solutions. This can actually be heated up to only 100 degrees C instead of 1,000 degrees C to purify the device again. When charging a battery, charged ions are inserted into one of the battery's electrodes. The researchers hypothesized that charging activated charcoal with chemical compounds called hydroxides would make it suitable for carbon capture since hydroxides form reversible bonds with CO2. This is exactly that thing that we were talking about, except this is going in deeper into the science of it. And so it says it's a new way to make these materials using a battery-like process. The rates of CO2 capture are already comparable to incumbent materials, but what's even more promising is that this method is far less energy intensive since we don't require high temperatures to collect the CO2 and regenerate the charcoal sponge. Because it's actually internally heated and it's not melting everything from the outside, it actually just kind of pops it off of the framework. The charged charcoal sponges developed by the Cambridge team only require heating to 100 degrees C from 90 to 100 degrees C. There's variability in the materials, so there's variability in the temperatures that can be achieved using renewable energy, whereas the older methodologies need natural gas and need to reach closer to 1,000. It says 900 C, but I believe the 900 to 1,000 C is what's needed. So to capture CO2 using the old methodologies, you have to burn natural gas. So you burn those greenhouse gases to try and capture greenhouse gases, whereas the other one can use solar energy.
I mean, that definitely sounds preferable.
Yeah, but like the other things, we were talking about it. What do you do with the carbon dioxide that you have? Iceland is throwing it into rocks, pumping it down into the rocks, and then when an eruption takes place or magma actually grabs that gas, it's gonna pull it out of suspension from the rocks. It's gonna melt the rocks. So why does that not pique somebody's curiosity that it's gonna turn a less than stellar eruption where it's just a fissure and some leakage into something that is gaseous and more explosive? I don't know if that's...
plus that whole release of carbon dioxide, right, which we're trying to avoid to begin with.
Yeah, well, I mean, a ton comes out from a volcanic eruption anyway. Why sit there and stuff a bunch into the rocks and then go, we're one of the most... We are a nation built from volcanic eruptions.
Like geothermal, yeah.
Why pump a bunch of CO2 down into the rocks and then wait for the eruption that's going to release it explosively? I must be ignorant of it, so I hope that somebody hears this and says, "Hey, Mayor Watt, we should talk." I don't know. I'll put you right over there.
Ring, ring.
Yeah. Oh my God. It's like, oh, hello, Iceland.
Exactly.
All right, let's keep going. This next one is going to be kind of odd. So over in ohmTown Daily is where the next article is. Artificial lymph nodes teaches T cells to kill cancer. Scientists have developed an artificial lymph node with the potential to treat cancer, according to a new study in mice and human cells. The newly developed lymph node, a sac filled with immune system components, is implanted under the skin. It is designed to act like a learning hub and stimulator to teach immune system T cells to recognize and kill cancer cells. What can take place, depending on the patient, is if they have cancer, at least one lymph node is usually acquired for testing to see if the cancer has spread into the lymphatic system. And if it has, then you have to be a little bit more aggressive in your treatment, like chemotherapy. Radiation treatment alone may not actually capture it all, so you have to go a little heavier handed. But if you can get healthy lymph nodes, and in another study we had, there was a researcher who converted other cells into lymph nodes. And so you basically had this whole mass array of lymph nodes all throughout the body.
That's right. Was that using liver cells, I think?
Yeah.
Or in the liver?
It was converting liver cells, into lymph nodes. So these lymph nodes, tiny glands throughout the body, mainly in the neck, armpits, and groin, are part of the immune system of mammals, including mice and people. They number in the hundreds so that immune cells in one body don't have to travel far to alert the immune system to take action. So Vanessa Wasta from Johns Hopkins put the article together for futurity.org. And to create the artificial lymph nodes, the scientists used it's such a weird sounding thing. I'm going to say this, hyaluronic acid, a moisturizing
H
substance.
acid.
Huh? Yeah.
I said H acid.
Yeah, H acid. Yeah. So you hear this a lot in marketing, that a moisturizer is hyaluronic.
Yes.
A moisturizing substance commonly used in cosmetics and lotions and found naturally in the body's skin and joints. Because of its properties, hyaluronic acid is often used in biodegradable materials such as wound healing patches meant to be implanted or applied to the body. Among those properties, hyaluronic acid, and this is the only article that I have ever read that says hyaluronic acid 15 million times.
Just making sure you get lots of practice saying it. Yeah. I'm glad you're the one saying it because I'm not sure I'm going to get it correct.
It can connect with T cells via a cell surface receptor. So basically, by creating these lymph nodes, these synthetic lymph nodes, it can teach T cells, hey, this is a cancer cell, go kill it. For the current study, the team used hyaluronic acid as the scaffolding or base for the new lymph node and added MHC, major histocompatibility complex, or HLA, human histocompatibility antigen molecules, which rev up T cells and other immune system components. Then they added molecules and antigens common to cancer cells to teach T cells what to look for. I'm always confused as to how they know, but I guess the antibody's reaction to a cancer cell is what identifies them.
Right, I guess that's kind of obvious, right? When you see it like going into complete destruction mode or something.
Yeah, exactly. So we have the ability to control what the T cells are being activated to search for, and lo and behold, they have 9 days later, mice with melanomas and colon cancers that received a combination of the artificial lymph node T cells and the anti-PD-1 drug had the best survival rates. 3 of the 7 mice were still alive at 33 days, compared to other groups that only lived for about 26 days. Not much, but it's still worthwhile to continue the investigation. This group of mice had the slowest cancer growth rate. It took between 5 and 10 days longer for their cancers to double in size than the other groups. The scientists also found that the artificial lymph node attracted an influx of other immune cells and acts as immunologically active niche to help to further stimulate the immune system. When T cells were injected into the mice alongside the artificial lymph node, T cell numbers grew as much as 9 times more plentiful. Which I guess is okay as long as the T cells don't go so wonky that they start taking out stuff that's good.
Exactly. I mean, it sounds like a really positive result, but that's the whole nature of cancer, right? Is like uncontrolled cell growth.
And let me see what they... yeah, I mean, if you have a bunch of T cells, then it's a sign that you might have cancer. Which I guess if you have an overabundance of T cells, it doesn't do any harm. But Livingston says the artificial lymph node approach is different from cancer vaccines, which typically activates a dendritic cell, an immune system component that teaches T cells what to search for. People with cancer often develop malfunctioning dendritic cells, and the artificial lymph node skips that dendritic cell to directly activate T cells. So, quite fascinating. Yeah, I don't know what else to say about this. I'm very glad that there are people out there that are doing this and have a grasp of just what the possibilities are. When I start reading about it, go ahead.
I was going to say, I think we are seeing a significant uptick in promising research about cancer, and I suspect a lot of it is because of technological advances, right? We can observe things more easily, we can crunch statistics more easily, etc.
Yeah, I just wish that things weren't so damn expensive, and that there was a real understanding that people don't have any bargaining power when it comes to this. I also don't like the idea of research where they pick and choose who it is that's going to be the patient to receive this based on the potential outcome being positive, because then it looks good on paper that they saved
Right.
somebody.
But then they don't actually develop something that's workable for the major population.
Yeah, or it might take longer. There are people out there that have cancers that they would, if they knew, then they would be, or if they were told that they have an option, they would be abundantly available. Here, I am here for you to, inject me with whatever it is. This gives me a chance, that kind of a thing. But it seems to be very narrow band. Only like one person gets it. And there's a whole ethics committee review and all of that kind of stuff that takes place. It's all a big paperwork thing. Oh, that's too bad. Anyway, let's go on to the next. Yeah, I know. Let's go on to the next article. The next article is over in Reality Hacker. The world's largest fungarium may unlock the mysteries of carbon capture. Research is uncovering the key role that fungi play in getting soils to absorb carbon, and how humanity's actions above ground are wreaking havoc on the mysterious fungal world below. Dun, dun, dun. This article, that is a dried mushroom, by the way. The article is over at wired.com. Matt Reynolds is the author. Research is uncovering the key role that fungi play in getting soils to absorb carbon. Eventually, Wired becomes a paywall. So I'm not logged in because I'm in the Arc browser. So at some point, it may actually stop showing. But let's just keep going. It says it's hard to miss.
Let's just chance it until then.
Yep, for sure. They say here it's hard to miss the headliners in Kew Gardens. The botanical collection in London is home to towering redwoods and giant Amazonian water lilies capable of holding up a small child. Each spring, its huge greenhouses pop with the technicolor displays of multiple orchid species. But that's not why we're here. Davies' own epiphany echoes a wider awakening of appreciation for these overlooked organisms. Mycologist Merlin Sheldrake's book, Entangled Life: How Fungi Make Our Worlds, Change Our Minds, and Shape Our Futures, was a surprise bestseller. In the video game and HBO series The Last of Us, it's a fictional brain-eating fungus from the genus Cordyceps that sends the world into an apocalyptic spiral. By the way, in as many days, I have heard references to cordyceps, and it's not like I'm seeking this stuff out. There was a video posted on Reddit of a spider that was infected with cordyceps and is in the last stages of its fungal overtaking. The cordyceps fungus actually takes over the spider or the insect and
This
causes
is
it.
like the zombie thing.
Yes, that's exactly what it is. Absolutely freaky to see. It looked like a face hugger coming to infect you. And if it would have landed on somebody, it would have probably given them a really nasty whatever. So the cordyceps fungal tendrils reach out from the soft gaps between an insect's limbs and then infect. And it's just nasty. Blah, 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 blah. So they say Martinez, whose research on mycorrhizal fungi, a large group of fungi that coexist with plant root systems. The mycorrhizal fungi form networks that go around and sometimes inside plant roots, transferring nutrients and water to the plant in exchange for carbon. Around 90% of the plant species are known to make these symbiotic trade networks with different species of fungi. These plants are covered by the fungi. It's incredible. They're small, but they're everywhere. This has serious implications for tree planting schemes, planting new forests, and a major hope for carbon sequestration. But there is increasing evidence that the mycorrhizal networks might be crucial to the success of these attempts. One replanting study found that a forest of birch and pine trees planted into heath moorland in northern Scotland did not increase soil carbon stocks even after nearly 40 years in the ground. The researchers who carried out the study think that it might be because the influx of new trees upset the delicate moorland mycorrhizal networks already present. Replacing the complete set of fungi with other fungi has implications for long-term carbon sequestration in soil and biodiversity. The fungi in polluted regions are less diverse, she says. This might have a knock-on effect on how well those forests store carbon. So we come in, trample all the ground, mess it all up, and it stops doing the sequestration, which means less carbon is pulled out of the air. Thus, we have climate change. So again, it's anthropomorphic because we mess around with nature. It's no longer as effective. Yeah, this is why we can't have nice things.
I mean, this, you combine this with some of the other articles we were discussing, right? Like we're going towards zombie plants with the photosynthesis, CRISPR, and I don't know. There's a lot of things we're tinkering with.
Tinker away, I suppose. Eventually we'll end up on Dune. All right, the last article for tonight, at least for Technology Today. Pushing past limits, tandem solar cells achieve over 20% efficiency. A tandem cell was constructed using perovskite and antimony selenide. This is the latest, whatever you want to call it, Wunder element, perovskite.
Oh, yes.
Type of crystal.
Yeah, I think I've seen it in ohmTown quite a bit.
Yeah. A research team has demonstrated for the first time proof of concept tandem solar cell using this combination. The article is over at SciTech Daily. Tsinghua, I think it is, University Press, which is from another article that we read tonight. This is the material. Researchers have developed a tandem solar cell combining antimony selenide and perovskite, achieving over 20% efficiency. I can't remember what current efficiency is. It's something like 8%. It's something really horrible. But I might be wrong. Maybe it's 12%. The device achieved a power conversion ratio of over 20%. The study shows that antimony selenide has great potential for bottom cell applications. Photovoltaic systems or technology which harnesses sunlight and converts it into electricity is popular because it provides a clean, renewable energy source. Did you find a number?
I did, but it's all over the place. Like I have one that's only 6% for silicon based, and then I have another average in the UK that's 15 to 20%. I mean, it's just, I guess it depends on the materials and
and then there's
variables.
longevity and toxicity issues and manufacturing issues. So they assembled a tandem solar cell with high conversion efficiency, using it as the bottom cell to demonstrate the potential of this material. Essentially, using perovskite and the selenium, they have the ability to capture twice as much, or an offset amount. So there's more on the perovskite and then less on the antimony side, allowing them to capture a little bit more of the overall solar rays. So enhancements to solar cell design, the team fabricated perovskite antimony selenide tandem solar cells that have a transparent conducting electrode for an optimized spectral response, and they were able to adjust the thickness of the transparent electrode array of the top cell to obtain a high efficiency of greater than 17% right off the top, and then they captured a little bit more on the bottom half. They optimized the antimony selenide bottom cell by introducing a double electron transport layer and achieved a power conversion efficiency of 7.58. So they actually ended up at close to 25%, which is, they should be leading with that, not 20.58%.
Exactly.
but they say it exceeded 20.58%. But that's already at, it's percentage. I'm sorry, strike that.
Oh, okay.
So maybe when it's combined, the efficiency comes out to being 20.58%, depending on the math. Like 7.58% of whatever came through, they captured 7.58 or whatever, or 3 point whatever. Because that's the difference. I mean, it's weird. 20.58 is not the difference between these. Like it lost 4%. I don't know.
Maybe, I don't know. We don't have enough data in front of us.
Yeah, I'd have to go and read the research study. So it's pretty neat. Unfortunately, I'm still in the camp that solar cells in times of peace are great, but they're not defensible by any stretch of the imagination. Not like oil, not like gas, not like even batteries are defensible, but what isn't defensible are things like solar and wind, even geothermal, hydro. They're not defensible. You can't protect them from threats to the extent that you can things like micronuclear power plants or thermoelectric power plants, or nuclear power plants, I should say. Those are defensible. You can bury them deep in the ground. You can put all kinds of shields, layer after layer of shield to prevent from a nuclear disaster taking place. But you gotta do it right.
I guess the only way you can have heavy reliance on solar is that if you have complete backup infrastructure.
Yeah, for sure. Yeah, I agree. All right, folks. But that's all 10 of the articles for the Technology Today show. We always kind of snap back to the very front of the technology. For each of our shows, we always come back to our home channel and then wish you all a good night. We've got one more show tonight called 4-Wheel Tech. That's going to take place in about, I'd say 10 minutes or so. So we'll get going. Just need to reset. And that means shutting down the stream, rebuilding that right there so that we can talk about some cars and technology. Sound good? Let's get going.
Sounds good.
I am Merwat, and right there is the visualizer for the sentient AI saying good night. If you don't stick around, good night.
Good night, hometown citizens. Be back shortly for 4-Wheel Tech.
See you soon.